Computational target discovery. The aims and objectives for this lecture are first to compare traditional, target-based and systems approaches to drug discovery. Second, to gain an overview of the target discovery process. And third, to understand the qualities of good drug targets. I've divided this session into 6 parts, and at the start of each part I will ask you to do a little exercise, go and look up some references, just to break up the session. So in the first part we will look at what a drug is, approaches to drug discovery, what a drug target is, and different types of disease and how the targets and drugs are going to change depending on the type of disease. In part 2 we will look at identifying drug targets and approaches to target discovery. In part 3 we will look at what makes a good drug target, targetability and drugability, and the importance of assays so that we can assess new targets and drugs, and the concept of going from hits to leads. In part 4 we will look at systems approaches, part 5 the idea of pathogenic deviations as drug targets, and finally in part 6 we will look at some examples and do an overall summary. So before we get started properly, the breaks between the different sections will be highlighted like this with a black background, and there will be an exercise for you to do. So the idea is that you pause the lecture at this point, go and do the exercise, and then resume the lecture. So your first exercise is to look at what you know about the nature of drug targets. What do you think you know about the different types of molecules that are drug targets? And secondly, what do you know about the interactions between a drug and a drug target? What types of forces are involved, and what do you think are the relative importance of those forces? So pause the lecture at this point and have a think about those different issues. Now we will start part 1 of the lecture, where we will look at what is a drug, approaches to drug discovery, and what is a drug target, and the different types of disease, and how that affects targets and drugs. We can define a drug target very simply as a natural molecular structure involved in the pathology of interest. And such drug targets can be proteins, such as receptors, where we have agonist or antagonist drugs that either activate the receptor or switch it off. They can be proteins like ion channels and enzymes. They can also be DNA in the case of things like cancer. And these days there's a new interest in RNA and ribosomal targets. But for the bulk of this lecture, we will really be talking about protein targets. Now, I asked you to think about the different types of interactions between drugs and targets. Now, there are 3 main types of forces that are going to be involved. First of which is van der Waals packing, just the interactions between atoms. So the concept of a van der Waals radius comes from the optimal distance between the centres of 2 atoms. Second, we have electrostatics, charge-charge interactions. Now these may be formal charges like a +1 on one atom interacting with a -1 on another, but they may also be partial charges. So in the majority of organic molecules the electrons are not evenly distributed, which means that some atoms have a partial negative charge or a partial positive charge. Thirdly, hydrogen bonds. So this is a hydrogen bound to an electronegative atom. Here we get a partial positive charge on the hydrogen interacting with another electronegative atom. In addition, we have the hydrophobic effect. Now, the hydrophobic effect isn't a force as such, but it is to do with entropy. I hope you're familiar with the idea of Gibbs free energy, delta G, which is equal to delta H, the enthalpy, which can be calculated from the three forces that I talked about previously. So delta G equals delta H minus T, the temperature, times delta S, the change in entropy. Now, when you take a hydrophilic atom or group and you place it into water, that hydrophilic group is making favourable interactions with the water. It is happy interacting with water, whereas a hydrophobic atom or group doesn't like to interact with water and therefore causes the water molecules to form an ordered kind of cage around those hydrophobic atoms. So that ordering reduces the entropy, and that means that our delta G, our Gibbs free energy, resulting from an interaction between the hydrophobic group and the water, is reduced. We don't have such a good delta G. Now, what do you think is the relative importance of these different effects? Because when you think about the interaction of any two entities, whether it's two drugs or two small molecules, a drug and a target protein, or indeed two proteins, you have to compare the interaction with water. So if you have molecule A and molecule B interacting with one another, you can look at the van der Waals packing, the electrostatics, the hydrogen bonds, the hydrophobic effect. And then you need to compare that with molecule A interacting with water and molecule B interacting with water. So if we think about van der Waals packing, well, water is very small. It can pack very well onto the surface of a protein, and indeed onto the surface of a drug. Electrostatics, well, electrostatic side chains, electrostatic groups, electrostatic ions are all very happy sitting in water. If that were not the case, then salt wouldn't dissolve in water, and of course we know that it does, because salt has a perfect electrostatic set of interactions with sodium interacting with chlorine in a kind of lattice. So why would you want to disrupt that if electrostatics are not happy being in water? Thirdly, we have hydrogen bonds. And of course, water really is the consummate hydrogen bonder. If water didn't hydrogen bond so well, then it would be a gas at room temperature. It's that hydrogen bonding ability that means that it is indeed a liquid at room temperature. So if you think about a protein and a drug interacting with water, well, all those interactions are just as good with water as they are with each other. So what we have left then is the hydrophobic effect. Now remember, this is the idea of a hydrophobic group being unfavourable interacting with water. So instead, if we can bury hydrophobics, then we get a very favourable interaction. So it turns out that an awful lot of small molecule drugs are actually very hydrophobic, because that means that they can optimize their binding energy. It's difficult with a small molecule to get a really good binding energy from the other types of interactions that we're talking about here. So if you conceptualize a perfect interaction between two molecules, they would have optimal packing, so the van der Waals energy is optimized, but they would also be completely hydrophobic. Now the problem there, of course, is that hydrophobicity does not give you any specificity. So those hydrophobic interactions could be occurring with anything else that's hydrophobic too. So what you end up having with a drug-target interaction is a trade-off between that really kind of optimum energy with specificity. Now what gives you the specificity? Well, the hydrogen bonds and the electrostatics and also the perfect shape matching. That provides you with specificity. And if all those things are optimally aligned, then you shouldn't lose any binding energy from that. But as soon as you get a mismatch, the energy won't be as good. The traditional approach to drug discovery and development has been very much physiology based. What I'm trying to emphasize in this lecture is the newer type of approach, which is target based. So a physiology based approach is really based around screening. In other words, we take a whole set of different compounds and we test them in animal models and cell-based assays to find out if they have any effect of interest. In a target-based approach, we're more interested in looking at the function of genes and gene products, and then taking a more design-based approach to finding a drug that will interact with that particular gene product, in other words, generally a protein, rather than just screening things at random. Now, as I mentioned earlier on, there are different types of disease which have different effects, and that means that the type of target that we're looking for and the type of drug that we're looking for will differ. So the first type of disease is related to genetic defects, and we'll come back to that towards the end of the lecture. The second type is related to infection, and that might be by bacteria, fungi, trypanosomes and viruses. And often there our drug target will be a protein that is expressed by one of those invading organisms, though not always. So very topical at the moment, a large number of the drugs being designed against COVID-19 or against SARS-CoV-2, the virus which causes it, are indeed targeted against the virus itself, mostly against the spike protein, but also against the protease, which cuts up the large protein which is initially generated by the virus. However, some of the drugs that are being designed for use in COVID-19 actually bind to ACE2, which is the receptor that the spike protein interacts with, and indeed to other proteins involved in the immune response to the virus. The third type of disease is the autoimmune or immune diseases, where something goes wrong with the immune system, and there the types of drugs which may be involved may not necessarily target the thing that is actually at fault. For example, in rheumatoid arthritis, one is producing autoantibodies. We don't target those. Instead, we target other things in the immune system to dampen down the immune response. The fourth type of disease mechanism is trauma. In other words, injuries, organ failures, and so on. And there, the drugs and drug targets that we're interested in may be related to removal of clots, or helping the healing process, or reducing swelling, or they may just be things that are related to pain control. And then we have multi-causal diseases, things like heart disease, cancer, all that type of thing, where we have really a plethora of different targets and different drugs that are of interest. What then is a drug target? And how do we choose a new target? So suppose you were working for a drug company of some sort, and you were asked to come up with a new target for their new blockbuster drug, which is going to make them billions of pounds. Well, there are two major routes to doing that. The first is to choose an established target. In other words, choose a protein that has been well studied and that we know that there are lots of drugs out there already. So for an established target, we have the advantage of a good scientific understanding of both the normal and the pathological physiology. There's drug industry experience, so we know that it's feasible to generate drugs against this target, and therefore our risk is lower. But although there's an understanding of the physiology, that doesn't necessarily mean that the mechanisms of existing drugs and exactly how they work is fully understood. So from a paper published in 2006, which I will ask you to download later, there were estimated to be fewer than 400 targets. Now, there's a newer paper looking at targets as well, and one of the things I'll ask you to do in the little exercise then is to look at that paper and see how that number might have changed. The alternative then is to start looking at new targets. Now, given that we know that the human genome contains approximately 20,000 proteins, and of course those also undergo alternative splicing, so on average we have about 3 splice variants per protein, so that means we have in total about 60,000 proteins. Given that this is the case, it seems an awful waste if we're only really addressing 400 or 500 targets. So by looking at these new proteins and how they're related to disease, we can expand our possibilities in terms of drug design. Now, of course, this is a much higher risk strategy than using established targets, but on the other hand, it has the potential for much greater returns. Looking back at the Overington paper that you will have a look at later, and looking at the detail of this, it turns out in fact that of those 324 targets that were in that paper, in fact only 266 of those are human and 58 are from pathogens. So actually we're only using 266. This, of course, was in 2006. We're only using 266 human targets out of 60,000 possibilities. Now, the number of drugs, as you see, is a fair bit larger. We have, at this stage, 1,204 small molecule drugs, 166 biologics, and 192 prodrugs. In case you don't know, a prodrug is a drug which, in the form that you take it, is not active, but it gets broken down in the body into an active form. A very good example of that actually is codeine. So the total number of drugs then in 2006 was 1,357. addressing 324 targets. So you can see on average each target has about 4 different drugs that is active against it. The other paper from 2008 gives estimates of fairly similar sorts of numbers, very slightly larger. And as I said, I'll ask you also to look at a more recent paper to see whether those numbers have changed significantly. At that stage, we're going to take another break. I'd like you to obtain copies of the following 3 papers and read the abstracts, and then focus a little more on the Santos paper, which is this more recent analysis of the number of drug targets, and look to see how those numbers compare with the numbers in the earlier papers. Okay, from the Santos paper, you should have found this table which details the numbers of new targets. You can see that the number of pathogen targets has increased hugely, and you can see that there has been a fair number of additional human protein targets as well. However, an awful lot of this is a result of a huge number of kinase inhibitors that have been developed, which actually bind to multiple kinases and therefore get counted as multiple protein targets. You should also have found this analysis, which is quite interesting, because the pie chart at the top left shows you the distribution of the major families of proteins that are common drug targets, and on the right the number of small molecule drugs that target these families. So you can see, for example, that GPCRs in the dark blue make up only 12% of the proteome, but 33% of drugs, so nearly 3 times overrepresented. Whereas the nuclear receptors, they similarly are hugely overrepresented. There are 3% of those in the proteome, 16% of drugs target them. On the other hand, looking at the kinases, that's 10% of the proteome, but only 3% of drugs. So the major difference, of course, though, is that 56% of protein families that are targeted by drugs fall into other categories, but only 30% of drugs. So this means that for these other categories, they are hugely underrepresented in the number of drugs that are being produced. Okay, moving on to part 2 properly now then, and we're going to look at identifying drug targets and approaches to target discovery. Now, this kind of flow chart tries to illustrate the different approaches to drugs and methods of identifying targets and identifying new drugs. So if we look in the middle at this region here, labelled A, the simplest kind of traditional approach is that we have a disease and we try lots of different drugs and we see what happens. Usually, of course, we're doing that initially in tissue culture or animal models or whatever. The slightly more complicated approach then is to go through a process whereby we're identifying a lead molecule. This is pathway B. We identify a lead molecule, we're trying to optimize that, we're looking to see how drug-like it is. We're looking at things like ADMET, that's absorption, distribution, metabolism, excretion and toxicity. We then go through animal studies, preclinical studies, clinical trials, formulation and finally producing a drug. Now, as we illustrate at the top right here, this is really a ligand focus approach. We're using medicinal chemistry, systematic screening, chemoinformatics approaches to do this. Now, the somewhat newer approach that I have been talking about is this approach C, where we have our disease of interest. We identify a target based on analysis of the actual disease mechanisms. We validate then whether that target actually has some sort of effect. We can do that with knockouts and knock-ins and so on. We characterize it in detail, and we then go into the ligand part. We identify a lead, we go through lead optimization and so on, eventually through clinical trials, formulation, and we produce a drug. Now, this is where we're starting to introduce this target-based focus. Now, the step up from that I will talk about a bit later, but this is to take a systems focus. So rather than just looking at the individual target and the individual pathway perhaps that it's involved in terms of what's going on with the disease, we're going to look in much more detail about things like disease models, metabolic pathways, flux balance analysis, all sorts of modelling which allows us to see whether there's maybe an alternative pathway. If you inhibit a particular enzyme, for example, is there another pathway that means that doesn't matter too much? We can also try to bring in ideas of drug resistance, particularly when we're looking at things like cancer and things like infection. So for the moment, we're just going to stick with talking about the D approach, in other words, the target-focused approach. So this little flowchart gives us an idea of the different steps that are involved in this process. We start with target discovery, so finding something that is involved in the pathology of interest. We move on to target validation, which, as I just mentioned, can be things like knockouts and knock-ins, looking to see whether if you do something to this particular target, it will have an effect on the pathology. Third, we move on to assay development. Having a good, reliable assay which allows us to detect whether a drug is having an effect is obviously critically important. We can then move on to screening, i.e. testing potential drugs or drug fragments, and that can be done in silico or it can be done in vitro. From there, we move from these initial hits to lead molecules, then on to lead optimization, drug development, clinical trials, and finally, one hopes, approval and market. Now, as you pass along this pathway, the costs at each stage become more and more. So the sooner you can curtail this development pathway, the more money you will save. So if you can knock out the idea of a target being important very early on, then you don't waste the money from lead optimization, development and trials. Depending on the type of disease that you're targeting, the approach to identifying drug targets may be somewhat different. So if you're trying to design an anti-infective, in other words, something that's going to kill a pathogen, then you want to look for targets which really have no human homologs. Because if you have human homologs, then when you produce a drug, then it's likely to interact with the human homologs as well, and therefore have side effects. On the other hand, if you're targeting a host protein, then ideally you want a target which has no paralogs. So I'll come back to the definition of paralogs, orthologs and homologs in just a moment. What you want to do then is annotate the sequence. You want to look for homologs with known inhibitors, homologs with known structure, the location of the protein, post-translational modifications, and so on. These are all important ideas because although in an ideal world one might find a host protein that doesn't have any paralogs, in practice, as we have seen already, there are lots of targets that are well established and have multiple drugs against them. And of course there are also well-known target families. As we've just seen, things like the G-coupled protein receptors are very well established as a family of proteins which are useful drug targets. And consequently, if we found a new drug target which is of interest but a member of that family, then we can use that information to model the structure, to find known inhibitors that we can then use as drug leads, and so on. Now, I said I would define the terms homology, orthology and paralogy. All this means is that if we have an ancestral gene or protein which then has evolved into two separate proteins, then those are defined as homologs. So the term homology simply means that two proteins or genes have descended from a common ancestor. Therefore, it is wrong to refer to percentage homology. Either two proteins or genes are homologous or they're not. Just as you are either dead or you're not, or pregnant or you're not. You can't be 15% pregnant or 37% dead. You can, of course, measure how long you have been dead for or pregnant for, and just in the same way, in principle at least, you could go back in time to see when these two proteins descended from a common ancestor. And of course, percentage sequence identity or percentage sequence similarity gives you an indication of how long ago these two proteins descended from a common ancestor. Now, the term ortholog means a gene duplication which has occurred as a result of a speciation event. So, for example, comparing our G6PD as humans, it's glucose 6-phosphate dehydrogenase, comparing the human version with the mouse version or the rabbit version or whatever. Those are all orthologs. Paralogs, on the other hand, are gene duplications within a species. So a typical example here would be the hemoglobin alpha and beta chains and myoglobin, all of which are related to one another. They're all homologous, but they're all present within one species. So I made the point on the previous slide that if you're wanting to identify a target within a host organism, so typically within a human, then you want to avoid targets which have paralogs, in other words, homologs within the same species, because that way you're likely to reduce the chance of an off-target side effect. But as I also said, we are often looking at big gene families like the G-coupled protein receptors, and we have drugs that target multiple members of that family. If we're interested in pathogen targets, then what we're really looking for is targets that are essential for the survival of that pathogen or for its virulence. So we might be looking for things that are involved in the biosynthesis of an essential metabolite, but if we do that, we have to check that there aren't alternative pathways that could have the same result. So a well-known example here is in trypanosomes. We have the protein dihydrofolate reductase, and this appears to be part of an essential pathway. But in fact, if we inhibit DHFR, there are other pathways that can often take over, so these inhibitors are not terribly effective as drugs. Also with pathogens, we have a need to be able to design a drug which ideally is broad spectrum. So while we want it to be specific in the sense that we don't want it to be interacting with other proteins and causing side effects, we do want it to work across different strains. So the target itself must be relatively conserved in terms of sequence across different strains if we're going to have that effect. Another approach to finding targets, particularly this time host targets, so human targets if we're talking about human drugs, is to use GWAS studies and linkage analysis. So these allow us to find markers that are linked to a disease. So this may be mutations, single nucleotide polymorphisms that are not actually causative of the disease in any way, but they associate with genes that are involved in the disease. This requires us to find the location of genes in the DNA and then to be able to annotate those genes if they're novel and we don't know what they're doing. In order to find the genes, we can use computational gene finding methods, but we can also use things like express sequence tags, where we're looking at messenger RNA and just sequencing the ends of it. But of course these days, with high throughput sequencing methods becoming so much cheaper, we can convert messenger RNA into cDNA and then do mass cDNA sequencing. Of course, the only problem with those sorts of experimental methods is that they are tissue specific. We're looking at the messenger RNA in a given tissue, whereas of course a computational method will not be tissue specific and we will just find all the genes. The problem, on the other hand, is that computational methods of gene finding actually aren't terribly accurate. Now, based on this idea of looking at messenger RNA, if we need to try to annotate a sequence in order to find out what it's doing, this is actually quite a complex computational problem. So if we're starting with a cDNA library, and then we're doing sequencing of ESTs, so as I said, that's just the first 500 or 600 bases off the end of a piece of messenger RNA that's been turned into a cDNA library, or indeed we are sequencing the whole of the cDNA. What we end up with from that is novel sequence data. Now, what do we want to do with that? We want to find out whether it's something of potential interest. So we may have found that it's associated with a particular disease, or we may find something that we haven't found before, and we just want to know whether it's likely to be important. So it's likely to be a member of one of these important families. So what we need to do then with our novel sequence data is do a blast run against a database like UniProt KB, where we have annotated protein sequences. And that then, if we get hits from that blast search, that allows us to annotate the sequence data. But of course it's all a bit more complicated than that, because we're going to get new data all the time. So we're going to want to rerun those blast searches whenever we have more sequence data. Similarly, the UniProt resource gets updated regularly. So when that happens, we want to rerun the searches again in order to reannotate any sequences that didn't hit anything in UniProt previously, or only hit with low confidence. Now, this type of approach has actually been used by a number of companies. In particular, there was a company called Human Genome Sciences, which has now been taken over by GlaxoSmithKline. They used this approach to find a novel member of the tumour necrosis factor family, and that actually ended up being a useful target for a disease called systemic lupus erythematosus, which is an autoimmune disease, and this essentially led to the first new drug in about 50 years. So this brings us on to the idea of functional genomics as a way of helping us to identify new targets. What we can do then, as I mentioned before, is cDNA sequencing. What we do is look at a tissue of interest from healthy individuals and diseased individuals. We sample the messenger RNA, convert it to cDNA. That's just done for stability, because messenger RNA tends to degrade. We can then sequence it in order to look at gene activity in healthy tissues versus diseased tissues. This allows us to identify genes that are involved in disease or are markers of disease. And they may also be involved in signalling pathways that are not part of the disease per se, but are again either markers, or if these signalling pathways lead to immune responses such as inflammation, then we can control the effects of the disease without actually interfering with the disease itself. So obviously we can use disease models in other organisms, and actually having a good model in another organism is really essential for being able to analyse what's going on with diseases and with drugs. We also would make use of high throughput techniques. I already mentioned sequencing, but also microarray techniques that allow us to look at gene expression in a very quick and rapid way. But now that sequencing has become so much cheaper, people tend to do that in a quantitative method rather than just using microarrays. And what we get out from all of that, of course, needs lots of statistical analysis. So at that point, we're going to have another break, and during this break I want you to have a think about what might be the good properties of a drug target. We've touched on some of them. I want you to think about that a little bit more. Take a look at this paper, What Makes a Good Drug Target, and you don't need to read the full paper, but it should give you some ideas to help you with your thoughts. In part 3, we're going to look at what makes a good drug target. We're going to look at the concepts of targetability and drugability, and also look at the ideas of assays, hits and leads, which I touched upon earlier. So what makes a good drug target? First of all, having a clear biological rationale for its use. Why is this particular protein linked to a particular disease or condition? Secondly, we want it to be specific to the pathogen or disease of interest. So when I say specific to the pathogen, of course what I mean is that it is not going to be a target that is also present in the human host. Of course, the importance of specificity is avoiding off-target side effects. So it's important to distinguish on-target side effects and off-target side effects. On-target side effects are those that occur when a drug interacts with a protein, somehow disrupts or enhances its function, and that has the good effects that we're looking for, but also has bad effects. On the other hand, an off-target side effect is when the drug interacts with something other than the target of interest, and it's those other interactions which cause the problems. Ideally, we want to ensure that there is no counterpart for the target in the gut flora. Don't forget that our guts contain billions of bacteria, and having a healthy digestive system means that these have to be present. So if you're producing an antibiotic, for example, that kills bacteria and also kills bacteria in the gut, then this can lead to all sorts of problems. The next thing that we need to consider is the drugability of the target. In other words, can a drug successfully interact with the target in the body? And finally, practicality, which largely comes down to assayability. In other words, is this target something that we can easily assay to say whether drugs that we're trialling are going to have a useful effect? So let's look at targetability. This is one of the terms that I want to define. This refers to the feasibility as a drug target. So is it specific to the pathological process of interest? In other words, is it unique to the pathogen as opposed to the host? Or at least does it have some level of discrimination from healthy processes? However, the word targetability can also refer to the ability of a protein to bind to a drug-like molecule. So, for example, things like hormones often are not targetable for small molecule drugs because they don't have pockets into which drugs can bind. Now, in other lectures that I will give you, we will talk about using antibodies as drugs, and in those cases the large shape of an antibody means that it can interact with convex surfaces or flat surfaces that normal small molecule drugs can't bind to. So that means that these sorts of proteins are targetable by antibodies but not by conventional small molecule drugs. The next point was target validation. We need to be able to demonstrate that a particular target has a critical involvement in a particular disease process, and that modulating that target in some way is likely to have a therapeutic effect. And I touched on this before, but typically we're talking about doing things like producing knockout or knock-in transgenic animals, or using these days RNA technologies in order to silence particular genes. And of course this also relies on having a good animal model for a disease of interest. Now, coming back to targetability, we can use computational analysis of protein function. Now this is going to rely on having protein sequence information or maybe protein structure information. Now sequence-based methods are relatively straightforward and easy because sequence data are readily available, can be obtained using high throughput methods and highly efficient methods, and these days very cheap methods. On the other hand, structure-based analysis has the advantage of being higher resolution because we can actually look at structural information, but obtaining the structure data is time-consuming and low throughput. Of course, if we have a homologue for which a structure is known, then we can use that as the basis for building a three-dimensional model of a protein. But of course then it is only a model, and we're basing analyses on a model which may not be correct. Drugability is another of the terms that I wanted to define, and this one actually has a number of somewhat different definitions that it's been given. First, however, it's how well a therapeutic can access the target. So that could include things like crossing the blood-brain barrier, absorption from the gut, and so on. Secondly, it's sometimes used to refer to the efficacy that a therapeutic can actually achieve when directed against this particular target. So parameters that influence drugability include the cellular location of the target. Is it on the cell surface, exposed on the membrane? Is it in the cytoplasm? Is it within the nucleus? And so on. Secondly, development of resistance, which is particularly important when we're talking about pathogen targets or cancer targets. Thirdly, transport mechanisms, and this includes things like transport of oral drugs across the gut, but also when we're talking about intracellular targets, it refers to import and export pumps that may bring the drug into the cell or remove it from the cell. And also side effects. We can't have a very therapeutic drug if we can only use it in very small quantities, and those quantities are not sufficient to have a good effect. So if we're interfering with a particular protein which leads to our solving our problem, but also leads to severe side effects, then that's clearly not going to be a very good drug. So one way of ensuring drugability, of course, is to use known successful drug target classes, as I mentioned before, such as the G protein coupled receptors. Now, the term drugability can also refer to the drug itself. So the idea of drug-likeness, compounds that are likely to yield useful leads. So I don't know whether you've come across it yet, but I'm sure you will before very long. Lipinski's rule of 5, which defines various characteristics about small molecules that are likely to make them good drug compounds. We can consider the drugability of a target by performing structure-based analysis. Now, we can relate fold to function and find functional regions, and then we can identify pockets. And of course this is part of the process of small molecule drug design in general. However, what we can also then do is screen those pockets for similarity to pockets seen on other human proteins. So this has been referred to as the pocketome. And in one paper, a comparison was performed of pockets in proteins on Mycoplasma tuberculosis, comparing that with the human pocketome, making over 245 million comparisons. And that is useful when we, in this case, are looking at producing some sort of antibiotic. But it applies equally to trying to avoid cross-reactivity when we're designing drugs against human proteins. Because here we're looking not just at homology of the overall protein, but looking to see whether individual pockets where drugs might bind look similar to one another. Now, I mentioned the importance of assays a couple of times. So when developing a good assay, there are a number of things that need to be taken into account. First of all, is the assay really relevant? Is it definitely an effect on the target of interest that we're measuring? Second, is it reliable and robust? In other words, can we reproduce it and get the same effects? And are the changes that we're measuring significant? Thirdly, practicality. This takes into account things like the time taken to do the assay, the availability of the reagents that are needed, how much effort it takes, and how much it costs to do. And fourthly, if possible, can we automate it for high throughput? So the next step, once we've identified a target, is to start looking for potential drug molecules. And this is somewhat outside the scope of this lecture, but still is part of the overall process, so I'm going to touch on it briefly. There are, of course, many ways of doing this, but essentially they fall into two categories. One is some sort of process of screening compound libraries, and this allows us to whittle down a large pool of potential hits into potential lead molecules. Now, typically this will be done in vitro using the assays that we've developed in the last step, and may also involve some counter-screening process for selectivity. So we screen for interactions with and effects on other proteins as well in order to try to avoid side effects. Now, in addition to being able to do those things in vitro, these days a lot of in silico screening can be done as well, particularly in the first stage of screening libraries of compounds against drugs, so basically big automated docking screens. The second of these two steps then is the idea of rational drug design, which is termed either computer-aided drug design, CADD, or structure-based drug design, SBDD. Now, this requires you to have a structure of the target, typically a structure that's been solved by something like X-ray crystallography, but it may also be a modelled structure. And what you can do then is to look at the interactions that your lead molecule is making and try to improve those interactions, to improve the specificity and to improve the affinity. This task of lead optimization then is one of refining the chemical structure in order to improve its characteristics. And this can be empirical, so we can do this by experiment as well as by computational methods, and of course even once you've used computational methods you're going to have to do actual experiments. It can also be combinatorial, so this is producing big libraries of slightly different compounds. And of course rational, this is the empirical tests that are based on our rational drug design. Another thing that we can look at is the activity of the drug and look at quantitative structure activity relationships, or QSAR. So what we're doing here is taking a lead molecule, making some changes to that lead molecule, assaying the activity of all those variants, and then projecting from that in order to suggest changes that would improve the drug further. Drug likeness is something that I referred to earlier, the idea of Lipinski's rules. So lead optimization may involve making changes that improve that, but we also have to worry about the pharmacokinetics and pharmacodynamics and the absorption, distribution, metabolism, excretion, and indeed toxicity related to the compound. Obviously the topics of screening, hits to leads, and lead optimization are rather outside the scope of this lecture. I've just touched on them to give you a feeling for the whole picture, but obviously in other lectures you will come back to those in much more detail. So at this point we're going to take another break, and what I want you to do is, using the information that we have just covered and the Gashaw et al. Paper that I mentioned earlier on, create your own flowchart of steps that you think might be necessary to decide whether a protein is going to be a good drug target. But also think about what other factors may have been missed out in defining a good target. And some of those I did touch on earlier, and we're going to come back to in more detail in the next section. Secondly, also obtain a copy of the Chandra 2009 paper referenced here, which is the basis of some of the things that we're going to talk about in the next section. In part 4, we're going to look at systems approaches. What we've described up until now has really been a single molecule reductionist approach. We've taken one target, one drug, one indication. So the idea with a systems-based approach to target discovery is that we take a much more holistic approach. So we look at modelling of networks, of pathways, and so on. And with such a system, we can take the idea of target validation through to in silico target validation by removing an individual gene or gene product within our systems model. Returning to the idea of anti-infectives, we can take a systems view by looking at things like network stability. So that allows us to understand how taking an element out of the network of interactions that occur between proteins, whether those interactions be direct or via metabolites, taking a protein out, how that will affect the network. We can look at conserved functions, phylogenetic profiling across hundreds of bacterial genomes. We talked before about looking at binding pockets. but we can do genome scale modelling and identification of pockets and comparison. And we can get functional clues from looking at what is expressed during infection and what is expressed during persistence. So we're going from starting with a systems analysis approach through sequence analysis, structural analysis and functional clues. So on the right-hand side, we're also looking at more complex things like metabolic fluxes, things that we've talked about before, like uniqueness, so non-similarity to the host genome and non-similarity to gut flora. Ideally, we're looking for broad spectrum targets. So in the case of Mycoplasma tuberculosis, we're looking for sequence comparisons across that family of pathogenic bacterial genomes, looking for things that are common to all of them, while also not being similar to gut flora and not similar to the host genome. Once we get to the functional levels, we want to start looking at non-similarity with human anti-targets. So this is kind of this reverse screening. We're wanting to make sure that even though there is no homologue present, that we're not going to bind to something else. And then we need to start thinking about things like target resistivity and the ability for the target to evolve a resistance to the drug. This extends the idea of network-based target discovery. This is a typical sort of network which, as I said, could be actual protein-protein interactions, or the interactions could be mediated by metabolites of some sort. So if we look at the top right, we talk about network analysis. So one of the characteristics of networks like this is that there tend to be critical nodes or hubs within the network, and then spokes coming off which have far fewer interactions. From this we can devise strategies for network disruption, identification of combinations of targets in some cases where you may need to hit multiple targets in order to disrupt this network effectively. And we're looking at the flow of information across the network and understanding how resistivity of a target can evolve. So looking on the left-hand side then, what we're trying to do is unravel new functional dependencies. So in functional terms, which are the key nodes in this network? And then we can start to look at disrupting this to understand the impact of gene deletions or inhibitions of the gene products, and whether there are alternative routes through the network that could mean that removing one individual node within this network is not enough. And then in addition to the metabolic networks, we need to understand how these things relate to signaling and information pathways and identify those. In networks, we often have this concept of robustness, and of course this is an essential property of biological systems. We want to be able to retain stability in the metabolism of an organism despite internal or external perturbations. So we have things like biochemical feedback, alternative pathways, and genomic redundancy. So for example, with phosphofructokinase, we have the liver, muscle and platelet versions of phosphofructokinase, and often if one of those is removed, then another one can kind of take over that role, maybe not quite as effectively, but can still fulfill the need. That was a rather short section, but we're going to break again at this point because I want you to have a look and see what you can find out on the internet about glucose 6-phosphate dehydrogenase deficiency. So this is an illness also known as favism or favism. So G6PD, have a look and find out what you can discover about this protein, what its function is, and what the result of deficiency in this protein is. In part 5, we're going to look at the concept of pathogenic deviations as drug targets. The idea of mutations causing disease and potentially being lethal was first potentially described some 2,000 years ago by Lucretius Caro, who said, "What is food to some men may be fierce poison to others." Now, of course, we don't really know what he was talking about, but it is possible that this was in reference to mutations in glucose 6-phosphate dehydrogenase, G6PD. So what happens here is that people suffering from this condition, when eating fava beans, or broad beans as we call them in the UK, and certain other foodstuffs, can end up with lysis of their red blood cells. And this condition actually affects up to 10% of the world population. It's caused by a faulty G6PD, which is a cytoplasmic enzyme coded on the X chromosome. Now, the function of G6PD is to, in this context at least, is to produce NADPH, which is used to regenerate glutathione that in turn reduces oxidizing agents such as hydrogen peroxide and maintains the reductive state of the inside of the cell. Now, in most cells, there are other ways of producing NADPH and regenerating glutathione. However, in red blood cells, because they don't have nuclei, the only pathway available for producing NADPH is this G6PD pathway. And that means that if your G6PD is not working properly, then it's much more difficult to maintain the reductive state inside the red blood cells. And these various foodstuffs, classically fava beans, but other things as well, and in addition a number of drugs, including antimalarials, lead to an increase in the sensitivity of red blood cells to oxidants. So what happens then is that the red blood cells lyse, and you get this hemolytic anemia as a result, which can result in really quite severe pain and, in extreme cases, death. So what I've hoped you've managed to find out about G6PD is that it is actually the most mutated enzyme in humans. And there are lots and lots of mutations known, and some of them are relatively common. So this first one, an A to G mutation at base 376, leads to an asparagine 126 to aspartate mutation, and this has very little effect whatsoever, and is present in 20% of African males. The 202G to A mutation, leading to valine 68 to methionine, leads to a small reduction in activity. About 90% activity remains, and that's present in about 20% of African alleles. On the other hand, the 563C to T mutation leads to serine 188 to phenylalanine, and that results in no activity at all. And that mutation is present in 20% of Mediterranean Caucasians. And as I said, there are many others as well, and they've actually been classified by the WHO into different levels of severity. Now, if we think about the effect that a mutation is going to have on a protein, we can describe three broad classes of mutation. Functional mutations, where residues that are critical for the protein function are altered. So this might be catalytic residues in an enzyme, something like that. We then have fold-preventing mutations. So these are mutations that stop the correct fold from being attained, perhaps because we have a small to large mutation in the middle of a protein and it just stops the protein from being able to fold up properly. And thirdly, we have destabilizing mutations. So these don't prevent the correct fold from being attained, but they raise its energy compared with unfolded or misfolded states. And these are the most interesting because, at least in principle, it should be possible to rescue protein function for the destabilizing mutations. Now, remembering that all proteins exist in an equilibrium between correctly folded and unfolded or misfolded states, that equilibrium is normally very heavily skewed towards the correctly folded form. So what a destabilizing mutation is doing is skewing the equilibrium the other way towards the unfolded or misfolded states. Now, if you can design a drug that binds to the correctly folded form without actually having any effect on function, then what will happen is that it will bind this correctly folded form and skew that equilibrium by the law of mass action over to the left in this diagram, meaning that we'll get more correctly folded protein present. So by doing this, we're going to achieve the desired effect of rescuing the function of a mutated protein. Now, you can think of that a little bit like a pencil. So if we broke a pencil, then we could wave that pencil around its break point, such that some of the time it was actually in the correct pencil-like shape, but a lot of the time it was in broken states. And what the drug is doing effectively is putting a piece of sellotape around the break and holding it in the right conformation. Now this concept means that we can rescue these damaged proteins, at least in principle. So as far as I know, there are no drugs yet that actually work this way, but certainly it has been shown that this could work. At this point I want to have a very brief break while you take a look at some papers that we're going to look at next in the examples. So again, just obtain the papers and read the abstracts, and then we'll come back to look at them. In this final part 6, we're going to look at a few examples and an overall summary. P53 is known as the guardian of the genome. It's a protein which is involved in detecting DNA damage, slowing the cell cycle to allow repair enzymes to do their work, and if the damage is too bad, then it triggers apoptosis, programmed cell death. So it's estimated that around half of human cancers have a mutation in P53, which then allows other mutations to accumulate and therefore lead to cancer. Now, one common mutation that is seen quite frequently in patients is also seen to be temperature sensitive. So lowering the body temperature during treatment, or at least lowering the temperature of the tumour before something like radiotherapy, has been experimented with as a way of improving the function of p53 such that the damage caused by radiotherapy or chemotherapy actually leads to death of the cells. Now, this fact that we get a destabilizing mutation confirms the concepts that I was talking about earlier, that if we get a mutation which simply stops or disfavors the correct fold of the protein, then there are measures that could be taken, such as lowering the temperature, in order to rescue that function. Now, the Jurger and Furcht paper that I asked you to download explores the possibility of stabilizing the structure into the active form by using a drug that would bind to the protein and hold it in that form. Our second example I did touch upon very briefly earlier on, and this is an example where one of these computational pipelines to annotate the function of proteins was used to produce a new drug. So this protein, variously known as BAF and BLIS, and it has some other names as well, is a member of the tumour necrosis factor superfamily of cytokines. It was found by this company called Human Genome Sciences by taking a human cDNA library derived from neutrophils and monocytes, so cells of the immune system, and they did exactly what I described. They set up a computational pipeline after taking the cDNA library and doing this EST sequencing. EST is expressed sequence tags, so it's just a case of sequencing the first and last 500 or 600 bases of cDNA copies of RNA. So they set up this pipeline, they screened these ESTs against Swissprot, and they eventually found this hit, which they thought was a member of this TNF superfamily. So having got this kind of tentative hit from the EST sequencing, they then sequenced the whole thing and found indeed that it was a member of this TNF family. And this is described in the Schneider et al. Paper. So obviously they patented this TNF family member because a lot of TNF family members have involvement in disease or could be useful targets for suppressing the immune system. Now, the patent actually listed a very, very long set of possible applications. And if you look at this paper by Fitt and Nodder from 2010, you'll see a bit of a description of the disputes that went on in court over whether this patent was valid or not. Because the thing is, when you have a patent granted, you have to demonstrate what's known as an industrial application for your invention. And in this case, they had such a long list of possible applications that it seemed very unlikely that a drug that targeted this TNF superfamily member could be useful for all of these applications. But it turned out that this ended up being the first new treatment for the autoimmune disease systemic lupus erythematosus in about 50 years. And the drug that they produced, belimumab, also known by the trade name of Benlysta, is expected to become a blockbuster drug, in other words, earning over $1 billion a year. To cut a long story short over the patent was eventually upheld, although originally in the High Court in London, it was found not to be valid, but it was eventually restored. My third example, and this is quite a brief one, is this paper by Tanaka et al. And the key kind of take-home point from this was that there was interest in developing a drug that would bind to this histidine kinase domain of the E. coli osmosensor called MZ in order to treat food poisoning from E. coli. Now, they tried lots and lots of different screening methods for drugs and really didn't come up with anything very successful. However, by doing very detailed annotation of this gene, this protein, by using a version of the BLAST software called SciBLAST, which allows for much more distant homologs to be detected, and doing very clever kind of searches in both directions with proper masking of the sequences in order to remove repetitive sequences and so on, which can confuse the BLAST software. They managed to detect homology with both heat shock protein 90 and DNA gyrase. Now, drugs against those two proteins were already known, and so these then were suggested as leads against the E. coli osmosensor, and indeed they did detect binding with these drugs, and that meant they could then refine these leads into trial drugs. So just to summarise then, the whole idea of target discovery for drug design can be done at many different levels. So starting at the bottom of this pyramid, we can start with gene sequences, genome architecture, protein sequences. So this is all sequence based. We can use sequence alignments. We can use these sensitive tools like SciBLAST, phylogenetic trees, comparative genomics, and so on. Going up a couple more levels, we can then start to look at protein structure and protein ligand complexes. So here we can be talking about using proper structures, ideally, but also models. We can then move into ideas like docking, molecular dynamics, identifying binding pockets, comparing those pockets so that we avoid side effects as a result of cross reactions. We can then start to move more into the systems area by looking at protein-protein interactions, looking at graphs of the sort that I showed you, looking at networks, and then integrating also biochemical pathways so that we can look at metabolic flux, metabolic control, and so on. And then we can move on from the simple in inverted commas, biochemical pathways into signaling pathways, transcriptional networks, the whole interactome, in other words, what interacts with what, and try to build up indeed a virtual cell. So this is really going into the depths of systems biology, trying to model what goes on in a whole cell. And above that, we can look at host pathogen interactions, modeling techniques based around agents, which is a way of doing machine learning, physicochemical shapes, and so on. And in our current situation with COVID, of course, the interaction between the virus, SARS-CoV-2, and the host cells is mediated by the spike protein on the virus, and the ACE2 surface protein on human cells. So an awful lot of the drugs that are being investigated for treatment of SARS-CoV-2 are focused on disrupting that host pathogen interaction. And then we can lead towards detailed disease models, where we're using these systems-based rules and simulations and analyses together with modelling at different scales in order to integrate all this information, try to understand where useful interventions can be made in a particular disease condition. In other words, where targets lie, and whether we might need to hit multiple targets at the same time in order to have a useful effect.